like to welcome everybody back to this afternoon's session. I knew I was going to feel like Bill Maher when I came out from the curtain, and I told it to my wife, and she said, "Don't do it." But it's a pleasure to be here and be moderating this first session of the afternoon. My name is David Stevenson. I'm the Senior Associate Dean for Maternal and Child Health. I'm also the director of the Johnson Center for Pregnancy and Newborn Services and the principal investigator of the Premature Research Center, which is a transdisciplinary center. We heard a little bit about transdisciplinarity this morning and its importance in solving some of the big problems in human health. This afternoon's session is focused on outcomes, not so much on prevention, which is what the focus of our center really is. And for that purpose, we have three outstanding speakers. The first speaker is Karl Sylvester. He's an associate professor of surgery in pediatric surgery. He's also the executive director of the maternal and fetal health program in the Center for Pregnancy and Newborn Services. He's an operating surgeon, so he's a real surgeon. But he's a special kind of surgeon in the sense that he's also looking to put himself out of business through the other kinds of investigative work that he does. And I know you'll enjoy hearing from him. Karl.
So thank you, David, and welcome back, everyone. Welcome to Stanford if it's your first time visiting the campus. I hope you enjoyed lunch. And as appropriate, my charge for the first part of this afternoon session is to talk to you a little bit about metabolism of prematurity. And we're going to go a little bit into the biology and the relevance of the biology that we've been able to make some observations about in the last couple of years, and whether there's any association and clues to some newborn acquired diseases of prematurity. So the population that I'm going to be speaking principally about is this population. This is a population of extremely low birth weight, or very low birth weight, less than 1,500 grams typically, and is born at a very premature time, less than 30 weeks of gestation. These babies, as a consequence of being born too early, are at risk for a number of acquired diseases, which are listed here in the lower half of the slide. And those include things that would affect the gut, principally, necrotizing enterocolitis, that would affect the developing lungs, known as chronic lung disease, and the early-going respiratory distress syndrome, that affect the developing brain as far as neurodevelopmental impairment and retinopathy of prematurity, which we heard a little bit about before. And while for a long time it's been recognized that one of the constant underpinnings of these four diseases of prematurity is inflammation, what is less clear and has been much less well investigated is what's the role of metabolism? And I would argue and try to convince you in the next 20 minutes or so is that upstream of inflammation actually is a metabolic reason that these babies get these inflammatory diseases of these quickly and rapidly adapting organ systems. But before we take the deep dive there for a moment, let's consider, for example, this is a global problem, and that the CDC reports that up to 25% of disorders related to short gestation and low birth weight are among the leading causes of mortality worldwide. And if you're fortunate enough to survive these austere environments, you're then faced with the problem of stunting, which is you continue to be too small for your gestated age post-conceptually, and the prevalence can be as high as 40% in austere environments throughout the world, as indicated by the burnt orange or the burnt red indicators. Now, the reasons for this is that essentially three principal problems. One is you don't have a steady access to robust food source. Two is you have a constant exposure to pathogens with frequent bouts of diarrhea. And in general, you're born into and live in unsanitary conditions. And together, these three things produce something called environmental enteropathy. And environmental enteropathy, if that's not bad enough for you, if not corrected or addressed by the age of 2, essentially locks you in to being stunted, very small for your size and for your age, as well as, perhaps more profoundly, never to reach your full cognitive abilities. What a big problem. Contrast that in the developing world, and this is what babies are born into in North America, and that is a neonatal intensive care unit. This is room 4 at Packard Children's Hospital at Stanford. And we've got all this technology and all this ability to monitor babies. Everything that you can imagine, and every microgram and microliter is measured. And yet, despite that, we still stick with technology from 1960. This little bag of yellow stuff here called total parental nutrition is a mixture basically of concentrated carbohydrate and proteins, and fats are administered separately as intralipids on several days a week. This was invented in the 60s by two surgeons, actually, at the University of Pennsylvania, Stan Dudrick and Jonathan Rhodes, and very little has changed since that time. In fact, we don't really measure for any kind of effectiveness of this therapy because it's considered nutritional. All we look for is signs of toxicity by measuring blood sugars and measuring some triglycerides from time to time. But what we don't understand is what's happening at the cell and the tissue level that might be predisposing some of these developing babies and these developing organ systems to the acquisition of diseases when these babies really should still be in utero. So we're going to proceed in the next 20 minutes under the basic hypothesis that intermediate metabolism of fatty acids, organic acids, and carbohydrates are really at the core of proper functioning of the mitochondria in each of our cells of these premature babies. And what's pictured in this cartoon is actually specifically the processing of acylcarnitines. So I have to explain a little bit of this because I'm going to be talking a lot about acylcarnitines, which are intermediates of fatty acid metabolism. So fatty acids can't be internalized to the mitochondria, which is the energy-producing source in most cells. They have to be esterified first through a acylcarnitine production. They then enter the matrix of the mitochondria, where they undergo beta oxidation, removing several carbons through a series of revolutions. But the bottom line is what we're after here is to produce intermediates for metabolism so that we can consume oxygen and produce energy. And when this system is not functioning properly, meaning it's not meeting the energy demands of a cell or a tissue, it produces toxic oxygen radicals. We've heard a little bit about this earlier in some of the talks. And it damages macromolecules. It damages DNA, it damages proteins, and it damages lipids. And when that happens, the cell is vulnerable to injury, which produces inflammation and ultimately acquisition of disease. So on the other hand, this is the environment where these premature babies need to be. And there's a lot of signaling taking place between mom and baby via the placenta. And what these signals are basically telling the baby is, what kind of environment am I going to be born into? What is mom's nutrition, and what's the influence of that on baby, and what can the baby expect to be exposed to postnatally? Now, this idea that fetal exposure to nutrition can have profound effects is basically accepted now and popularized, because if you search the Barker hypothesis, you get this wiki page, which basically explains that if a baby is exposed to austere or low nutrition in utero, there's biologic adaptations that take place that prepare them and put them at significantly increased risk for the development of acquired diseases as an adult that include things that we can typically consider as metabolic syndrome: obesity, hyperlipidemia, coronary artery disease, stroke, and diabetes. And the reasons for that may be is that you were born not expecting to see a lot of nutrition, and then you grow into adulthood where food is abundantly available, it's cheap, and you don't have to work to obtain it. You actually store it because you were really prepared to be born into an environment where food is not available to you. And this effect is so profound that you can either be undernourished or overnourished in utero, and it not only affects the F1 generation at the mitochondrial level, but because the gametes and the germ cells of that F1 generation has been affected by the same exposure in utero, it affects the next generation. It passed on through oocyte and sperm to the F2 generation. So you can see where a cycle that would be quite vicious of poor nutrition would result in significant impairment and problems. And as I told you, when it involves cognition, you can see how socioeconomic status might lead to some trapping. Okay, let's return for a moment back to the neonatal intensive care unit and dive a little bit deeper into the role of metabolism. So for the remainder of the talk, I'm going to focus on this paradigm disease, necrotizing enterocolitis. This is true and close to a pediatric surgeon's heart, and as Dr. Stevenson said, I'd love to put myself out of business and never operating on one of these babies again. This disease almost exclusively affects premature babies, less than 1,500 grams. It has a prevalence up to 10% in those that are of the smallest and youngest ages, and it occurs in two varieties. One, it exists only in the mucosa, which is the inner lining of the intestine, and it resolves. Two, it involves a full thickness, where the intestine dies and has to be removed by a surgical procedure. If you're lucky enough to survive, you still have a 25% incidence of severe neurodevelopmental impairment that is lifelong. And the cost of care here is enormous, estimated between $500 million and $1 billion in the United States alone. Now, the big problem with this disease is we understand very little bit about its etiology, and therefore, we're unable to prevent it, we're unable to predict it, and we actually diagnose it quite late, which leads to all the downstream morbidity and the increased mortality of 50%. What we do know is that there's specific risk factors for necrotizing enterocolitis, and they're almost exclusively and strongly associated with this, and that includes a background of prematurity, the presence of a baby who is being fed, and you have to have an intestinal lining that has been colonized. And we're all born with a sterile GI tract, and over the first couple of days of life, the microbes come in because we need them, and they're able to help digest foodstuff. And in the course of digesting foodstuffs, they ferment and produce various gases, like methane, like hydrogen, that when present in excess produce the X-ray that you see on this slide, which is a finding of what's called pneumatosis intestinale. And pneumatosis intestinale, when present, suggests the intestine has got a problem, and that on the inside, we've got a real problem in that it's dying. This is necrotizing enterocolitis, and these are the fundamental problems. What we don't understand is how all these things happen in such a short period of time, and why can't we predict them with any greater futility? So in order to address this problem, we've taken a reductionist view, and that is we said, let's look at available biologic samples in human beings, because a lot of the rodent models that we were seeing in the literature just didn't seem to make sense to us. We also took biologic samples from repositories, and we matched those with available clinical data on large population data sets. We made some observations, we saw some molecules that we were able to model on a cellular and a subcellular at the mitochondrial level, and then build back up through an animal model that we felt was relevant and apply it again back to human beings. And this is the story where we are today. So one of the first experiments that we did was a transcriptomic analysis, where you basically take tissue from the intestine, and you grind it up, and you look at some of the transcripts, which is basically what the DNA of the cell is telling them how to execute what kind of biological program. And we compared babies with necrotizing enterocolitis who actually had developed the disease to another inflammatory bowel disease that has bimodal onset in children and adults, ulcerative colitis and Crohn's disease. And when you looked at what were the genes and the transcripts that were being expressed in these cells that were different, almost uniformly, we always saw the same thing, and that was there was metabolic pathways. There was branched chain amino acids, there was organic acids, and there were some other amino acids which were very curious. And what this all reminded us of, and what it really sounded quite familiar to, was newborn screening. These same metabolites that we were seeing in babies with necrotizing enterocolitis are searched for on every single baby who's born in North America and in many developed countries. An inborn error of metabolism is a very rare disease. It's a single gene defect typically that results in a vulnerability for a disease and end organ damage. And we screen for these things by doing a Hill stick to put it on a blood card and send it to a state laboratory where mass spectrometry, a fancy machine, identifies various metabolites that are irregular. It's not diagnostic, it's screening. But the reason it works is because if you identify a baby who is at risk, you can prevent the onset of the morbidity by pure alteration of some of the diet. Obviously, that's oversimplified, but that's the general concept. And we said there seems to be some real similarities here between what we're seeing in those babies that have necrotizing enterocolitis and what's looked for on newborn screening. We're talking about at one time the genetic disease, and on the other hand, an acquired disease. What's the similarity? So this was a flower plot, and what we did next was we annotated all those metabolites that are looked for on newborn screening, and we attached those to genes that were either known because of common variants that led to a variability and an inborn error metabolism, or a rare variant that was now available and told us that a slight variation in the level of metabolite might be associated with acquired diseases like necrotizing enterocolitis. We also then said, where can we get a lot of information? So we were very fortunate to partner with the state, the California Department of Public Health, and the Genetic Diseases Screening Program to obtain data on over 2.6 million live births, of which approximately 160,000 of these were premature. And we were also fortunate because these births were linked to a discharge summary and a birth certificate. So we knew exactly what happened to all of these infants. And the question we asked was, because of all these other observations, is there any association or pattern that we could find in newborn screening that was associated with the subsequent development of necrotizing enterocolitis? Now, bear in mind, newborn screening is done within the first 48 hours of life, and necrotizing enterocolitis is something that occurs several weeks into life in premature babies. We were astonished when one of the first observations we found was that there was global metabolic dysfunction when we looked at the levels of acylcarnitines. And we looked at this at an unbiased view, where we said, let's throw out the thresholds that would suggest you're at risk for an inborn error of metabolism, and let's just let the data speak to itself. And nearly every single acylcarnitine that we looked at had a clear association when it was abnormally regulated with necrotizing enterocolitis, as demonstrated by these adjusted odds ratios. And the adjustment was for gestational age and birth weight, importantly. We then said, well, that's pretty interesting. So could we use this to predict or forecast who is most likely to get necrotizing enterocolitis, or to enrich a population of babies that we would want to put under intense scrutiny, just like happens with newborn screening? If you're abnormal and you're at risk for a genetic disease, you undergo strict guidelines for feeding, as well as ongoing testing. And what we're able to develop is, through a reduction of features to just 6 acylcarnitines, including age, weight, and the presence of TPN, that stuff that's in the yellow bag, we could produce a model that would find 90% of the babies that would ultimately get necrotizing enterocolitis. And importantly, this model was equally effective, and if not more so, in the really premature baby, less than 32 weeks, but even more effective if you were a little bit older, 32 to 36 weeks. And the model performed best in the first 48 hours of life before confounding by clinical care. Clinical care would certainly affect the values that we're measuring in the blood. More on this in a minute. Perhaps even more interesting was, everything that we do around preterm babies is typically risk stratified by your age and your weight. And what we found was, and demonstrated on this slide, is that if you took out the age and the weight, and you just use the acylcarnitine profile that we were able to measure, you could essentially replace agent weight for risk stratification, suggesting that the biologic profile that we're seeing with the metabolites was the surrogate. It could tell you what was the inherent biologic susceptibility that replaced agent weight. So a lot happens between when you're born, several weeks pass, there's a lot of clinical care, there's a lot of different exposures, and the onset of necrotizing enterocolitis. The earliest babies, those born between 24 and 28 weeks, typically get it after a month of life, and those born a little bit later, 28 to 32 weeks, typically get it after 2 weeks of life. And these exposures, we said, what happens in those babies that actually have necrotizing enterocolitis? And so for this study, we took advantage of a consortium that I ran for several years with Larry Moss from Nationwide Children's Hospital, where we had been collecting blood samples on every baby at the time of diagnosis. And the time of diagnosis would be that X-ray finding that I showed you, pneumatosis. And we took those blood samples and we exposed them, or we conducted the same mass spectrometry analysis that you use for newborn screening in those babies that now had NEC. And again, what we found was not necessarily surprising, but exciting. And that was there was a lot of amino acids and acylcarnitines that significantly and statistically associated with necrotizing enterocolitis that when taken together suggested that these babies had severe mitochondrial impairment and there was a metabolic acidosis taking place that we should be able to find before that fact occurs. So that's part of the equation in that we feel that if there's metabolic dysfunction and you've got this accumulation of metabolites, but what's going on at the level of the mucosa where all the action is? So in order to understand this, we said, this sounds a little bit like predisposition biologically and environment are conspiring to produce this disease, much like we started with when I talked about environmental enteropathy. So we said, perhaps there's a newborn enteropathy that we should be looking for. In order to look at this, we said, again, everything is happening as a result of metabolism and this interaction at the gut. So let's go a little bit closer and model this in an animal model where we chose 14 and 28-day postnatal mice. Now, there's a lot of reasons morphologically and functionally why we chose those, but suffice it to say that a 14-day postnatal mouse is akin to a second trimester baby, and a 28-day postnatal mouse is akin, at least from the gut perspective, to a term baby. And we exposed the guts of these mice to varying concentrations of something called butyric acid. Butyric acid is the most abundant short-chain fatty acid that is produced by the fermentation of carbohydrates and the interaction with microbes in our gut. It's a carbon-4. And when we did this in a dose-response curve, we consistently and uniformly showed that the 14-day mouse, as depicted on your left side, histologic assessment, showed a lot of cells that were sloughed and dead. The mice and the intestines were much more sensitive to this. And in fact, the blue streaks that you see there are all the proteins that were being eluted out in the mouse poop. And one of these proteins, which is labeled ZZ at the bottom there, really caught our attention because it seemed to be consistently qualitatively and quantitatively associated with this injury that we were seeing as a result of the fermentation products, in this case, butyric acid. It's also important to note, and we asked the question, well, before you even give any feedings, is there anything differently metabolically about function in a 14 versus a 28-day mouse? And as it turns out, when you isolate mitochondria and you determine how much oxygen can they consume and how much ATP can they generate in response to the typical clues like ADP, there's more than a 50% reduction, as showed by the histogram here, in what a 14-day mouse is able to do at baseline than a 28-day mouse. And as you will recall, I told you that if your mitochondria can't respond to the demands for energy, reactive oxygen species are produced, macromolecular damage ensues, and you get tissue-level organ damage. Now, why were these cells dying? We had some thoughts about this. There's things called necrosis. There's things called apoptosis, which are manners in which cells die. There's also something called necroptosis. And necroptosis is kind of an alternative pathway that is just now beginning to be investigated and involves two very important proteins, RIP1 and RIP3. And in response to a phosphorylation event, which is just a post-translational modification of a protein, they assemble into a complex that actually attacks the mitochondria, opens some pores, and results in the destruction or the death of the cell, typically in response to either inflammation or a metabolic challenge. And what we found in the mice was that in response to butyric acid, we had an accumulation of the RIP proteins, as depicted in the top bars here. And equally importantly, we had an abundance of reactive oxygen species, as demonstrated by this 4-HNE product, which is 4-hydroxynonenal. And basically, that shows you that there's protein damage as a result of oxidative stress. So things are starting to come together to say, yes, perhaps butyric acid, we have a mechanism of how it's causing the cells to die at the local level in response to a metabolic deficit. In addition to that, we found that ZZ protein, in this case, we've now termed it NECZ, was specifically located to the enterocytes in the mouse intestine, that it could be produced in response to butyric acid, and it actually could be removed, and you could prevent necroptosis through small molecule antagonists to necrostatin, which is one of its inhibitors. And perhaps more astonishingly, and we were very pleased to see, that this same protein could be found in the stool in those babies that developed necrotizing enterocolitis. And it was independent of other inflammatory mediators, which is usually the shortcoming of biomarkers for this typical disease, and in this case, calprotectin. We looked at those babies that had high and low levels of calprotectin in their stool and in their bloodstreams and found that this marker was highly specific for the onset of necrotizing enterocolitis and independent of inflammation. This work was done with KT Park, one of our gastroenterologists here, and we're interested, and we have some other work around this inflammatory bowel disease for another day. Perhaps more interesting than that was that this same protein was associated with recovery from necrotizing enterocolitis. Now, when we're dealing with these babies in the intensive care unit, we kind of say, I don't know, maybe it's 10 days, maybe it's 14 days of antibiotics. Maybe we feed them on day 20, maybe we feed them on day 25. And now what we were able to see is that this protein clearly demonstrated a recovery of the intestinal mucosa and the enterocytes back to a healthy state. We divorced it from the necroptosis pathway that was killing the cells and tells us it's okay to reintroduce enteral feedings, which is really what the babies need in measured doses. So taken together, what I've told you is that newborn screening, which is a measure of systemic metabolic fitness used for inborn errors of metabolism, perhaps can be repurposed to look for acquired diseases that have a metabolic basis. And perhaps we've now been able to develop a stool assay that we can link to that metabolic impairment that's occurring at the level of tissue injury in the gut for the disease necrotizing enterocolitis, which provides the opportunity to say we could possibly triage babies by newborn screening for acquired diseases, and then we could monitor them further for the development of disease by a simple stool test. And so taking this all together, the way we've continued to conceptualize this is that I think that there's probably a developmental susceptibility because of prematurity that metabolically is associated with this metabotype, we call it, of our phenotype of what your metabolites are. And when you compound that with an environmental risk, like our neonatal intensive care unit, which at first blush you would say is not austere at all, but with all these different exposures, which are not normal for a premature baby, are in fact producing perhaps diseases like necrotizing enterocolitis. And if you're lucky enough to survive that, you're still left with a baby who has diminished growth potential and significant impairment of neurodevelopmental outcomes. So clearly, preventing this disease would be of the utmost importance and interest. So the current working hypothesis is that perhaps prematurity is associated with metabolic dysfunction, but as Dr. Stevenson likes to challenge me and ask me, particularly in the March of Dimes, is perhaps it's the other way around. And perhaps metabolic dysfunction is actually one of the causative factors that's producing prematurity. This, too, is a talk for another day, but it's intriguing to think about it this way. And finally, if nothing else, I think what we're really excited about is that you've got this population-level data of metabolomic data. And we've been able to produce, at least at the first blush, a test that looks like it might help us for a triage tool identifying the highest risk babies for necrotizing enterocolitis, possibly for sepsis. What if we applied it to chronic lung disease, which we're going to hear a lot about? How about the need for energy neurodevelopmentally? And finally, how about the onset of childhood and adult onset diseases, which is what our metabolome might be able to tell us in those very first days of life, which reflects the exposures that occurred in utero. So with that, I'd like to close, acknowledge some of the people that did all the work that is behind all of these crazy ideas, particularly people from my lab, Bruce Ling, G. Tao, Zach Kastenberg, Tiffany Sinclair. Collaborators across the country at leading children's hospitals have been part of the NEC consortium that allowed us to do this. Dr. Laura Jaleef-Pulowski at the California Genetic Disease Screening Program, now at UCSF, and researchers at Stanford, including our own David Stevenson, Gary Shaw, Tina Cowan, Greg Enns, and many others. So with that, I'll close, and thank you for your attention.
Thanks very much, Karl. Welcome to the new generation of surgery. And now you know what I meant when I said that this is a surgeon who's trying to put himself out of business as a surgeon with one of the real scourges of the nursery, which is necrotizing enterocolitis. We have time for some questions if people would be interested in asking any of Karl.
No, I mean, I don't think anyone would argue that there's clear benefits, and of all the things that have been tried, breast milk for sure, probiotics possibly as preventative of things like necrotizing enterocolitis. This is a complex problem. It's multifactorial. That's pretty well established. Interestingly enough, despite the increased use of breast milk, NEC has not gone away. And in our unit, actually, we have a remarkably low level of necrotizing enterocolitis. Now, we use a lot of breast milk, but I think that's not the whole answer. And part of the answer, it might be rate of feeding, which is also well established, but also responding. Every baby's different, and we've said it for many years. You could have 10 bassinets in a row. These babies could be of all the same gestational age and of all the same postnatal age, all exposed to the same environment and breast milk, and yet one or two of them get it and the others don't. And why is that?
I think also, Karl, you focused the last part of your talk on necrotizing enterocolitis, but you began your talk with a reference to a global problem related to enteropathy, stunting, and chronic disease. And that is a problem of huge magnitude for the world. And associated with preterm birth, it's 5%. It's the main cause of death less than 5 years of age, I think, in the world right now. So these kinds of investigations are extraordinarily important.
Yeah, thank you. I mean, one of the problems, as you know, is that we don't have good tools to objectify at the molecular level risk, particularly metabolic or molecular risk. And I think newborn screening is an opportunity for that, particularly when you're talking about environments where you want to do a study that are austere. I mean, all you need is a heel stick and a blood spot, and it doesn't even have to be frozen. It can be mailed. And we go off to a laboratory where we can risk stratify babies and try to project what's their risk in an austere environment because of environmental enteropathy for these things like stunting and neurodevelopment or cognitive impairment.
And you can imagine scaling these kinds of screens to the global setting in communities where you can identify particular individual babies that might be at risk, and then directing your attention to those infants with various kinds of intervention. Any other questions people have? I have one more. You mentioned that I've challenged you several times about the fact that you might flip this around, and rather than looking at it as some kind of maturational description, that you're actually beginning to identify those pathways that might be relevant to the pathology which creates a preterm baby and some of the phenotypes that we see in the nursery.
Yes.
You want to comment further about that?
Yeah, so this is intriguing, and it's sometimes hard to get your brain around, but thank you for that comment. I agree. I mean, at first blush, you'd say, well, it's obvious. You're developmentally arrested because you're premature, and therefore your metabolism hasn't quite worked yet. And yet he says, well, what if it's the other way around? And I don't have a good answer, but I will say this. I think that it's going to be related to inflammation again. So we've talked a lot about, and you did in earlier sessions, about why is it time to go, right, when it's prematurity? Why is it time to get out of the womb? And perhaps it's an energy mismatch, which produces the inflammation. And that's why, through my own biased prism, everything is about metabolism. But I'm interested in talking to you and Brees and some others in the center about that relationship from a molecular signaling standpoint between metabolism and inflammation.
And we're talking to people all across this university about these kinds of things because we don't want to leave anybody out as we think about a complex human health problem. So I think this gives you a sense of how transdisciplinarity can work. We have surgeons interested in preterm birth and in these kinds of complications, not just with innovation in the operating room, which he does also extremely well, by the way.
You're very kind.
And we appreciate him as a surgeon when we need him for that purpose. Any other questions? Okay, Karl.
Thank you very much.